The multiplication rule can be used to predict the number of gametes that an individual is going to be able to produce. If we look at a very simple example, if we have something like this, there's one gene there, the A gene that we're looking at. And for this gene, notice that there are two different A's. There's a capital A or a lowercase a. So that means that this individual can produce two different gametes. Now, obviously, that's a very simple example, but it is important because we take the same concept and we apply it when we're looking at something much bigger. So if we have something, for example, like this. This individual, if we wanted to predict how many gametes this one is going to make, if we use the rule of independent assortment, that means that what happens with the A gene is not going to affect what happens with the B gene. And so since it is independent assortment, then we can use the multiplication rule because that deals with independent events. So if we look at the A gene, just like before, we have two versions of A present, so I'm going to put a little 2 there. And for the B's, we have two versions of B present, so I'm going to put a 2 there. And independent events, we can take them and we can multiply, so I'm going to get 4 different gametes being possible for this one. Another example, if we had something like this, so if we just make something where we're actually looking at 4 genes. In this case, we have an A gene, a B gene, a C gene, and then a D gene as well. We look at each one of them separately. The A gene, we've got 2 different A's, so I'm going to put a little 2 there. For the B's, both of them are capital B, so while there are 2 B's, they are both the same version. So since that is the case, I'm going to put a 1 there for my B's. For the C's, I have a capital and a lowercase, so that's 2 different ones. And then for D, I have 2 different ones there. And we do the same thing, we're just multiplying straight across. Make sure that you do multiply and you don't add here. And so when we multiply across there, 8 is going to be the total number. So there's 8 different gametes that would be produced by that particular parent. So that's just a look at how many different gametes we can get from an individual. We could take the same idea and we could use it to predict exactly how large a Punnett square might be if we had a cross and we wanted to know whether we were going to go ahead and draw the Punnett square or whether we wanted to use probability rules for it. If we wanted to know how big is the square, so by square I'm talking about the Punnett square, well, we have to know how many gametes we have from each parent. So if we had something like this, and we were crossing it with this one, if we go through and we predict how many gametes from each one, for this one there's 2 different A's, 2 different B's, and 2 different C's. Again, multiplying it, we're going to get 8 different gametes from that parent. And then this one over here, we're going to have 2 different A's, 1 type of B, and 1 type of C. And if we multiply that, we get 2. So if we were to actually draw this Punnett square, it's going to be an 8 by 2. 8 by 2 means that there will be 16 boxes total in that particular Punnett square.